Hello, I am Sam Smith, and this is Sam's Mcat Basics. This podcast covers the most important topics put out on the AAMC Mcat each year, and I determined this list by going through all the official practice materials that the AAMC puts out, and also some third-party practice material, and just put together this big list of topics that consistently kept on showing up, and ranked those topics and made podcasts out of it. This podcast is the second podcast in a series that covers genetics. This podcast is going to cover three main topics. The first of that is going to be different inheritance patterns that you'll see in the MCAT. The second is going to be gene expression, which is a pretty high yield topic on the MCAT. And then the third is going to be different genetic laboratory techniques. So I'm going to talk about FISH. I'm going to talk about the different blotting techniques. I'll talk about PCR and a few different forms of PCR that you may not be familiar with, things like qPCR or real-time PCR. And yeah, so I hope this all helps and you get something good out of it. So as I said, I want to get started with inheritance patterns. So the first I'm going to talk about are sex-linked traits. And so in some cases, a gene is present on the X chromosome. And so when the gene is present on the X chromosome, its inheritance pattern is going to be a little different than when it's inherited, through when it's an autosomal trait. And so I'll go through those next. These X-linked genes or X-linked traits are different than autosomal traits because these genes are present in different copy numbers in males versus females. As I talked about earlier, males have one X chromosome and females have two X chromosomes. And so genes on the X chromosome can either be recessive or dominant. And when a gene is recessive on the X chromosome, it means that females need two copies of this recessive allele, i.e., their two X chromosomes both must carry this recessive allele. Whereas for men, they only carry one X chromosome. So if they have that one recessive allele, they're going to have that phenotype. They're going to have that, disease usually is the way you look at it. It's important also to remember that for men, they receive their X chromosome from their mother. So as I was saying earlier, you receive each chromosome from your mother and your father, and so for your sex chromosomes, you receive, if you're a male, you receive your Y chromosome from your dad and your X from your mom. So for a sex-linked recessive disease, if you're male and your mom is afflicted with some sex-linked recessive disease, you are automatically going to get that disease because you're inheriting her X chromosome. That's going to be your only allele. It's going to be the only allele for that gene. On the other hand, if you are a female, you're receiving two X chromosomes, one X from your dad, one X from your mom. If your mom is afflicted with some disease, you might, and say the disease is, again, sex-linked recessive, you're not necessarily going to get that disease because your dad, might have a different allele for that disease, and you might be all right. You might not have a mutated allele or something like that. So these sex-linked diseases tend to typically afflict males more than they do females. Before I give you guys a quick example here, I want to make it clear that I've been using sex-linked and X-linked kind of interchangeably. However, some diseases are not necessarily X-linked that are sex-linked. You can have diseases that are linked to the Y chromosome. They're much more rare, and you can kind of think through those problems sort of like you would an X chromosome. Obviously, females don't get a Y chromosome, so these diseases would only affect males. But in terms of the MCAT, I think these X-linked diseases are much higher yield. To illustrate how these X-linked diseases affect men and women differently, I'm going to go through kind of an example, phrase it like an MCAT question, and this example is red-green color blindness. Red-green color blindness is a X-linked recessive disease. And so, say you have a mother who is afflicted by this disease, and you have an unafflicted father. Now, what is the frequency of children born that are male and affected and that are female and affected? So to solve these problems, what I'll do is I'll draw out a Punnett square, and then you can do the little simple math even in your head and figure it out. I'm not going to go through how to draw a Punnett square just because I don't think having it described to you is really going to be very helpful. But go through, watch a YouTube video if you don't understand. If you do, great. So after I've drawn up a Punnett square here, you can see you're going to have 100% of the sons that are born will have red-green color blindness. And none of the daughters will have red-green color blindness because they're saved from this disease by their father's X chromosome that's functional. And it's always important to tie in different sociological theories, sociological topics into these discussions. So this is where I tie in discrimination. So obviously here is a form of discrimination by red-green color blindness. In the case that affects more men than women, a very significant form here of gender discrimination. Now, all jokes aside, this is just to show that these X-linked diseases affect men and women differently. A lot of times they're much more prevalent in men because men only receive one X chromosome. Another inheritance pattern is called autosomal inheritance, and so this is a little bit simpler. You'll have autosomal recessive and autosomal dominant phenotypes. And so for recessive, you have to have two copies of this recessive allele. And for the dominant, you only have to have one of these dominant alleles, then you're fine. You're not affected by that autosomal recessive disease. And in this case, it's a lot like the women in the X chromosome. You're getting two copies, one from your mother, one from your father. You can look back, again, use a Punnett square to figure out what the percentages are of children you'll have that are affected and unaffected. And so, a lot of these questions on the MCAT are going to basically just ask you, calculate the frequency that two specific parents are going to have, a child that's affected with some autosomal recessive or autosomal dominant disease. And a lot of the times, they might throw in, what are the chances that it's a boy that's affected or a girl that's affected? Or they might do two traits in which you got to do a dihybrid cross, or even a trihybrid. I think I saw on one practice test. But anyway, so these questions are pretty easy. Just, this is something you got to practice and get down, but once you do, it's not too bad. And real quickly, one thing that really did help me solve these problems pretty quick was to memorize the monohybrid and the dihybrid phenotypic ratios. So a monohybrid cross refers to a single gene. So it's one gene that you're looking at with this monohybrid cross. And so the ratio for mating two heterozygous individuals is 3-1, i.e., that is, 3 people will have the autosomal, or will have the dominant allele, and 1 will have 2 recessive alleles. So it's important in figuring out, how many people are going to be affected with a autosomal recessive disease. That's 1 in every 4 people, right? Because you have 3 plus 1, that's 4. 1 in every 4 will be affected. And then, so the ratio to remember is 3-1, i.e. there's going to be 3 people who carry at least 1 dominant allele, and there's going to be 1 individual who has 2 recessive alleles. For a dihybrid cross, which refers to 2 different genes, you're going to get a phenotypic ratio of 9-3 to 3-1. And so the 9 refers to there's going to be 9 individuals who carry both dominant alleles. So they'll carry a dominant allele A and dominant allele B. Let's just say the 2 genes are A and B. And then the 2 3s are going to correspond to the people that carry, a dominant allele of gene B, but no dominant allele of gene A, or the reverse, where you have the dominant allele of gene A, but not of gene B. And then the 1 is going to refer to that one individual that carries all recessive alleles. And so, again, this is pretty easy. It's going to ask you, these two genes, you have these two genes, the parents are both heterozygous. What is the frequency of individuals who carry all recessive alleles or have, this disease? And so here you can say, okay, you have 9, it's going to be 1 to, you add up all the numbers, that's 9, 10, 11, 12, 13, 14, 15, 16. It's going to be 1 out of every 16 individuals carries this disease or has all recessive alleles for the two genes. All right, now that I've talked a little bit about these basic inheritance patterns, let's bring it a step further and talk about some of these less known inheritance patterns, things like genetic imprinting. So genetic imprinting or genomic imprinting is an epigenetic phenomenon that causes genes to be expressed in a parent of origin specific manner. So that is to say that you inherit from one of your parents. You directly, whatever your parents express, either one of them, you inherit that phenotype specifically. As I talked about before, we usually have 2 copies of a certain gene, right? One from your mom, one from your dad. However, for genetic imprinting, one of these 2 copies is completely just shut off. And this happens in a parent of origin, as I said, parent of origin dependent manner. So this shows up as either maternal inheritance or paternal inheritance only. There's no substitute allele, which makes imprinted genes more vulnerable to the negative effects of mutations. So, to just kind of illustrate this, you'll have an affected father. Then if you have an affected father and it's an imprinted gene, all the children will be affected. This is, of course, paternal inheritance or paternal gene imprinting. And then you can have maternal gene imprinting, where if your mother is affected, she's going to pass it along to all the children. And that's because one of these chromosomes or one of these genes is just completely silenced, and you're only expressing a certain gene from one chromosome. And obviously this inheritance pattern looks very similar to sex-linked traits. However, for gene imprinting or genomic imprinting, you'll see every single child affected. If the mother is affected and the father's chromosome is silenced in all the children because of this epigenetic modification, then you'll see every single child affected. It's not just going to affect 25% of the females, 50% of the males, or you're not going to just see the males inherit this genetic disorder, but then the females in the family not, that are the children. You're going to see every single child affected, regardless if they're a male or female. So right now your head might be spinning, you might be thinking, how exactly does this happen? So there are 2 main factors that play a role in genetic imprinting. The first is DNA methylation. So DNA methylation tends to silence genes. So this can happen in a parent of origin matter, especially for genetic imprinting, right? So, you can inherit a set of genes from your mother or father. Your mother's genes can all be methylated, and therefore you only express the genes that you inherit from your father. The other factor that plays a role here in genetic imprinting is histone modification. So again, histones, they are proteins that help compact DNA, and in this case, these histones can also be modified, and they are modified by methyl groups. Again, they're methylated, and this can act either way. So this can open up the chromatin, which encourages gene expression, or it can close off chromatin, which will actually decrease gene expression. And this is very dependent upon where this methylation takes place. So it's important to note here that in the case of histones, they're proteins, and when they are methylated, it's the actual amino acids that make up the histone protein that are methylated. In the case of DNA methylation, it's the actual DNA that's methylated. All right, so I just wanted to touch on that topic briefly. Let's now move into a completely different topic and talk a little bit about genetic linkage. So if you recall in the last podcast, I talked a little bit about Gregor Mendel, a little bit about his experiments. He came up with the law of independent assortment. And basically the law of independent assortment states that the inheritance of one pair of genes is independent of the inheritance of another pair. That is, if you inherit one set of genes, you are no more likely to inherit another set of genes because you inherited that set of genes. They won't affect each other. However, genetic linkage is the main exception to this rule or law of independent assortment. So genetic linkage is the tendency of DNA sequences that are close together on a chromosome to be inherited together during the meiosis phase of sexual reproduction. And so what makes genes linked is their actual physical proximity to one another. And it is measured in recombination frequency. So what is recombination frequency? What does that term mean? Well, homologous recombination refers to the crossing over event that occurs in meiosis. So if you remember from the last podcast, in meiosis, prophase 1 of meiosis, you have crossing over where these chromosomes meet up and then they swap various portions of their chromosomes. And as you could imagine, genes that are more closely located to each other are more likely to be crossed over together to the chromosome. They're more likely to stay together when recombination occurs. So, recombination frequency then is the frequency in which a recombination event happens between two genes. So, it doesn't necessarily measure the frequency that two genes are crossed over together, or two genes migrate, to another chromosome together during crossover. It's the frequency of a crossover event happening between two genes. And so this number ranges from 0% to 50%. So for 50% means these genes are really far apart, and these are unlinked, independently assorted genes. And then for 0%, these genes are literally right next to each other, and these crossover events really never happen between these two genes. So the big takeaway here is that recombination frequency measures how linked genes are, and a very high recombination frequency of 50%, which is the max, means that two genes are unlinked and assort independently, and then a low recombination frequency means that genes are linked and that recombinations never occur between those two genes because they're so close. And I really wouldn't worry too much about the kind of complex calculations involving genetic linkage and recombination and building these maps. I wouldn't worry about these calculations that you might have seen, in genetics class or whatever, because I don't think I ever saw any of them on the Mcat or any of the practice materials. I think these problems would probably take too long, and therefore they're left off the Mcat. All right, so let's move into gene expression now, which is a very important topic, and I'm going to cover how gene expression is regulated in transcription, translation, and how that works for prokaryotes and eukaryotes. And then I'll talk about DNA repair enzymes, and how those work. Gene expression can be regulated at a few different levels. It can be regulated at the transcriptional level, translational level, and also a little bit during replication, actually. And so I'm mainly going to talk about gene expression controlled at the transcriptional level because that's the most common, but I will also touch a little bit on gene expression control at the translational level as well, protein modifications, and how that happens. So if you remember from, basic genetics, transcription is the conversion of DNA into mRNA, and then translation is conversion of mRNA into protein. So let's begin with talking about transcriptional control in prokaryotes. When you think prokaryotes, think bacteria, and so prokaryotic genes are often found in operons. And so these genes in an operon are transcribed as a group, and they all have a single promoter. And I like to think of an operon as, a van full of people that are carpooling to work. The driver is driving them, he's controlling all these other people. They're all individual people, and but there's a driver. There's one person who's controlling where all these people are going, and so the operon controls a certain set of genes. That could be 5 different genes, could be 10, whatever. But that operon controls the transcription of that set of genes, and they're usually related genes. So, for the sake of this example, let's just say it's a van full of a family, and they're all related. And so these operons contain regulatory genes and structural genes. Regulatory genes code for proteins that regulate transcription or act as binding sites for other regulatory proteins. And structural genes encode for the proteins that are not regulatory. So these are the proteins that are actually functioning within this prokaryote. So, say an enzyme or something like that. And regulatory proteins control how much of the operon is transcribed. And they do this by binding to the DNA itself. And there are a few types of regulatory proteins. Again, remember, these regulatory proteins are produced by regulatory genes. So there are 2 main types of regulatory proteins that you're going to want to know, and you're going to want to know what they bind to. So the first is called a repressor, and this binds to a piece of DNA called the operator. And when it's bound to the operator, it reduces transcription, it represses it, which makes sense. And the other type of regulatory protein you're going to want to know is called an activator. And an activator binds to part of the DNA sequence called the enhancer. And as you could probably guess, when the activator binds the enhancer, it increases transcription of the operon. And it does this by helping the RNA polymerase bind to the promoter. So the promoter region is another region of the DNA or of the operon to remember, and the promoter is where the RNA polymerase binds. It's also important to note that the operon may be inducible or repressible. So an inducible operon is an operon that is always turned off, and it is turned on by an inducer, which is just some kind of small molecule that binds it. And so you can think about an inducible operon as like a car sitting at a red light. It's always off, and then your inducer molecule is a green light, and that's what starts your car going. That's what gets your car on the road. And then you also have a repressible operon. And so this is an operon that's always on, and then it's turned off by a co-repressor, which again is some kind of small molecule that binds to it. And this is like a car that hits a red light. So this car is going, it's on the road, it's good, boom, boom, boom. It's hit by a co-repressor or a red light, and it slows down and must come to a stop. And remember that everything I just talked about only applies to prokaryotes. Eukaryotes are a little more complex. I'm going to talk about that next, but just know that operons apply to prokaryotes and are very, very rare in eukaryotic cells. All right, so on to transcriptional control in eukaryotes. And as I was saying, this is like 10 times more complicated than bacterial regulation. I think a little less common on the Mcat from what I can remember, but still important to know. So in eukaryotes, you have transcriptional factors. And so these are proteins that bind to enhancers or silencers in the DNA and affect transcription. And so transcription factors must have a DNA binding domain. Remember, this is a defining feature of transcription factors, DNA binding domain. Take the example of chromatin remodeling. So chromatin remodeling involves the opening and closing of chromatin to allow transcriptional machinery to access the DNA. And again, we have euchromatin, which is open chromatin, and this tends to have higher levels of transcription, therefore higher levels of gene expression. And on the other hand, we have heterochromatin, which is tightly packed DNA, which has a lot less transcription, if any at all, and less gene expression. And so things like histone acetyltransferases, histone deacetylases, and histone methyltransferases modify histone proteins in order to either open up the chromatin or close off the chromatin. However, these factors are not transcription factors, and that's because they don't contain a DNA binding motif. Instead, they bind to or modify histones and affect gene expression through that route instead of going and directly binding to DNA. So remember that, very important. Just because some protein or some molecule influences gene expression does not mean that it is a transcription factor. Instead, it must have a DNA binding domain. All right, so now back to the region of DNA that these transcription factors bind to. Again, these are called enhancers and silencers. And so enhancers increase transcription when they're bound, and silencers decrease it. And the main difference in eukaryotes compared to prokaryotes in this facet is that enhancers and silencers can actually be very far away from the promoter, and they can be either upstream or downstream. And the DNA actually forms this kind of loop. It forms this big loop back on itself, and so that the transcription factors can be bound to enhancers in their silencers, and this bend causes them to actually be in contact with the promoter. So if you can imagine a long snake, pretend the head is the promoter region, and then the tail is the enhancer and silencer. And so how do you make these two come in contact with each other? Well, you take the snake's head and its tail and you bend it together and you touch them together. This is essentially what happens in order for the enhancer or the repressor, or the enhancer or the silencer to be in contact with the promoter itself. And so again, the promoter is where the RNA polymerase attaches and then transcribes the DNA into mRNA. And in this bend, you're going to have transcriptional factors that bring these two regions of the DNA together, and then you're also going to have mediator proteins that perform this function as well, that don't directly bind to DNA, but bind one transcription factor to another and hold this bend together. So important thing to remember about this is that just the DNA folds over on itself, and these transcriptional regulators that are these distal control elements, enhancers or silencers, they're far away from the promoter and must bend in order to get in proximity and exert their control of either increasing or decreasing gene expression. Another important form of gene expression regulation comes in the form of post-transcriptional modifications. And so these only happen in eukaryotic cells, so don't think this happens in bacterial cells. And there are three main post-transcriptional modifications that you're going to need to know for the MCAT. The first is intron-exon splicing. The second is the 3' poly A tail, and the third one is the 5' 7-methylguanosine cap. So introns are part of the mRNA transcript that are removed, and exons are part of the mRNA transcript that stay in the mature transcript. And so it's kind of backwards. It's kind of hard to think about introns you would think would be in the transcript, but they're actually out. So just think about it backwards. So introns are removed, exons stay in. And this can happen in different combinations. This is called alternative splicing. So, one transcript you might have the intron 1 removed, and in the other transcript you won't have that piece of DNA removed, in which intron 1 becomes an exon. And so by doing this, you can change the transcript and can control gene expression. A particular example of alternative splicing is called exon skipping. In this mode, a particular exon may be excluded in mRNAs under a particular set of conditions or a particular tissue, and then this exon is omitted from the mRNA in others. So that's just called exon skipping, which refers to a particular exon of interest. So the second post-translational modification I want to talk about is the addition of a 3' poly A tail. So the poly A tail is added obviously to the 3' end of the transcript, and it is important for nuclear export, translation, and also stability of the mRNA. So a longer poly A tail leads to a longer degradation time, and therefore this transcript can float around in the cell for longer, and more of it can be translated into protein, so that affects gene expression. And the last instance of post-transcriptional modification that I want to talk about is the 5' G cap, or the 5' 7-methylguanosine cap. And so again, this is at the 5' end of the transcript, and it protects against degradation and is very important in being able to be recognized by translation initiation machinery. So this is what helps the ribosome recognize the mRNA transcript. So real quickly, I'm just going to summarize the gene expression regulation that I've just talked about. So prokaryotes have these things called operons, which are these groups of genes that all have a single promoter, and they're controlled by repressor proteins, which binds to the operator. And then you have activator proteins, which bind to the enhancer, and they kind of do what they sound like they do. Repressors repress, activators activate. And it's important to remember that these operator enhancer genes are very close to the operon, or part of the operon. And for eukaryotes, you have enhancer and silencer regions of the DNA that transcription factors bind to and either increase or decrease transcription. And it's important to remember that these regulatory factors can be kind of distantly far away from the promoter, and in order to be in close proximity, the DNA can form these loops. And in both cases, for eukaryotes and prokaryotes, you have these promoter regions where the RNA polymerase binds. So now that I've talked or briefly touched on transcriptional regulation and regulation of gene expression, let's get into DNA repair. So there are three main types of DNA repair mechanisms that you need to know for the MCAT, or at least this is the three that I think you need to know. The first is proofreading, which is done by the DNA polymerase. Then there's base excision repair, and there is mismatch repair. So I'll go over how each of these three works. DNA proofreading basically does exactly what it sounds like it does. The DNA polymerase, which is the replication enzyme, is able to check its work as it goes along. So if it recognizes a mistake, it can then remove that mistake by removing the incorrect base that it put into the DNA, and it can then replace that base with the correct base. Now, if you remember, DNA replication occurs in the 5' to 3' direction. So, in other words, the DNA polymerase travels from the 5' end of the DNA to the 3' end. However, the proofreading function of the DNA polymerase actually reads from 3' to 5'. And so each DNA polymerase has a 3' to 5' proofreading exonuclease domain in the actual protein itself. And so this allows the enzyme to check each nucleotide during DNA synthesis and then excise or kick out the mismatched nucleotides. And again, they do that in the 3' to 5' direction. All right, so now moving on to base excision repair. So Nature, the very prestigious journal, describes base excision repair as a process that removes and replaces damaged or misincorporated bases in one strand of the DNA that may generate mutations if left unrepaired. The damaged base is modified and excised to create a single-stranded DNA break that is repaired to regenerate an intact helix. In other words, you have some mismatched base, and that base itself is just excised out of the DNA, and then you repair it. Pretty simple. And there's actually a really large cast of enzymes that are responsible for this base excision repair. The protein or the enzyme that recognizes these mismatches are DNA glycosylases. And then AP endonucleases cleave these sites in order to remove that base. You also have DNA polymerases that are able to add in single nucleotides, and these, of course, are a little bit different than the DNA polymerases that go the length of the DNA during replication. And then lastly, you have DNA ligase, which might sound familiar because it's an enzyme that is used in replication. And DNA ligase catalyzes the sealing of the gap between this new nucleotide that the DNA polymerase added and strand of DNA that had been mutated, or the strand in which the mutated base was cut out of. So it just seals that gap and creates a contiguous piece of DNA. So the prime example of base excision repair is when uracil is found in DNA, right? Because shit ain't supposed to be like that. Uracil is supposed to be in RNA only, while thymine is the base that's in DNA. So just to give you a brief example, so let's say you have a uracil in your DNA. Well, the first step in base excision repair is that the uracil base is excised, leaving an abasic site, or a site that has no base. And then the second step, your AP endonuclease removes the abasic site, i.e. the backbone that was left over after that uracil base was excised. And then, step 3, you have DNA polymerase and DNA ligase come in. DNA polymerase repairs that base, puts in a new one, and ligase seals a gap, and boom, your DNA is repaired. You protects against these mutations, eventually might cause cancer or some other disease. All right, so on to the last DNA repair to know, and it is mismatch repair. And so mismatch repair is a system for recognizing and repairing errors in either insertions, deletions, or misincorporation of bases that usually happens during DNA replication and recombination, as well as repairing some forms of DNA damage that happen from like UV rays or reactive oxidative species, etc. And so this repairs mismatches like, when a G pairs to a T or a C to an A, or any other combo that is a mismatch. So the enzymes for this process are a little bit more complex, complicated. I'm not going to go through those. Just know that they are called mute proteins, M-U-T, mute proteins. And there's a bunch of different ones, and they form dimers, trimers, and through these kind of complex processes, do the following. So the first step is a mismatch is detected in newly synthesized DNA. And once that's detected, the new DNA strand is cut, and the mispaired nucleotide and its neighbors are removed. So you remove a big chunk of that DNA, and then the missing patch is replaced with correct nucleotides. This is done by the DNA polymerase. And then the DNA ligase seals the gap between that base or the bases and the backbone. So what is the biggest difference between mismatch repair and base excision repair? And I think this is pretty important to understand. Mismatch repair cuts out a big chunk of bases. So you cut out the mismatch and a bunch of its neighbors. And then the base excision repair removes a single base and replaces that single base. Other than that, they're very similar. Of course, they have different enzymes. And it's important to note that mutations in the genes that code these proteins can result in cancers, because then, you mutate your mute S protein in the mismatch repair system, and, now you're not able to repair these other mismatches, and your mutations start to accumulate, and eventually you can develop cancer somewhere, and cancer metastasizes and can lead to death, etc. Not good. All right, so now that I've covered all of these different DNA repair mechanisms in detail, let me quickly recap. So the first DNA repair system is the proofreading system. And so the DNA polymerase is able to recognize these mismatches as it goes along, if it makes a mistake, and it can remove and replace these bases, and this occurs in the opposite direction that the DNA polymerase is traveling. The next is base excision repair. That's where you remove a single base and then replace that base. And then you have the most common, which is mismatch repair system. And this will actually remove a bunch of bases around your one mismatch and then fill that back in. All right, on to the last topic for this genetics podcast. And this is kind of genetics. I kind of threw it in just because I thought it was pretty important. But this is the genetics laboratory techniques. And, this stuff showed up a bunch on the practice material and even on the Mcat. So, well, allegedly it showed up on the Mcat. I don't think I'm supposed to talk about it. But nonetheless, let's get it going. The first laboratory technique I want to talk about is PCR, or the polymerase chain reaction. So this is a widely used technique in molecular biology to amplify a single copy or a very few number of copies of DNA in order to generate a bunch of copies of that. So this is an amplification process in order to increase the number of a specific DNA molecule. Most people in the lab would use this if, say, you have a rare gene and you want to check it out, from one cell, sequence it from one cell, compare that to a bunch of different cells. Well, you're not going to be able to sequence just this gene if there's only one little molecule of it. You're going to have to amplify that and then sequence. So, there's a lot of different applications, but the process is the same for PCR in all instances. So you have an initial phase called the denaturation phase, then you have an annealing phase, and then an extending phase. In the denaturation phase, you heat the DNA up to about 95 degrees Celsius, and the DNA strands separate. And then once they're separated, you lower that temperature down to 55 degrees Celsius, and this allows the primers to bind to the DNA strands. This is called the annealing phase. And in the last phase, which is called the elongation phase, you increase the temperature to about 72 degrees Celsius, and your polymerase binds to the primers, and then it extends and adds bases along the length of that DNA. And again, if you think about it, these molecules are still separate, and they have those primers in there, and this just allows the polymerase to run through and add bases onto that DNA, which is single-stranded at the moment. And so it's important to note that one PCR cycle, which is these 3 steps I just described, increases the amount of DNA you have by 2, right? Because you're breaking apart one strand and you're creating 2 new strands. So you're doubling the amount of DNA in one cycle. Also important to note that these temperatures are kind of subject to change. So, different enzymes are going to operate at a different temperature. So this is going to change the elongation temperature, and then you can change up the times. There's a ton of different optimization you can do within PCR. And something important to understand for PCR and for all of these techniques is, what do you need in order for this reaction to happen? What chemical components do you need? Well, you need, number one, a DNA template. So this is what you put in that you want amplified. And you need a DNA polymerase enzyme. You need primers, which the DNA polymerase recognizes, and these are usually pretty short sequences, maybe 20 sequences long or 20 base pairs long, and these bind to the DNA when it's been opened up. Then, of course, you need these nucleotides, right? Because you need something for your DNA polymerase to add to these opened up strands. And then you need a reaction buffer. And, buffer just makes sure that these enzymes work, that nothing degrades. And so how do you choose the right primer? Well, primers are these short oligonucleotide sequences, 8 to 60 base pairs in length. And if your purpose of your project is to randomly generate sequences or randomly amplify sequences, they can be whatever. But a lot of times you must know these sequences that you want to amplify because you need to design primers in order to bind to these regions that you can then amplify. You may also see the term qPCR or Rt-Pcr, and I'll talk about those in a sec when I talk about different types of blotting, but these are for studying gene expression or quantifying gene expression. All right, so let's talk about blotting. So what you need to know about blotting is essentially what the different blotting techniques are used for. You don't really need to understand the procedure, why is it called blotting? I would just ignore that. Just know exactly what these things are trying to look at, what information you're trying to get from them. So a Western blot is used to detect the presence of a particular protein in a mixture. The probe that is used is therefore not DNA or not RNA, but antibodies, right? Because antibodies can bind to specific proteins. And this technique is also called immunoblotting because your immune system uses antibodies. So, Western blot, proteins. All right, moving on. So, the Southern blot is a technique for detecting specific DNA fragments in a complex mixture. This technique was invented in the 1970s by Edward Southern, and it has been applied to detect restriction fragment length polymorphisms and variable number tandem repeat polymorphisms. And you might be saying, what the hell are those? Or you might have a good understanding of what they are, but just know that these can be used to identify individuals. Everybody has their own, different pattern of restriction length polymorphism, and each individual has a different number of variable tandem repeats within their genes. So again, the Southern blotting is used to detect specific DNA fragments in a mixture. All right, on to Northern blotting. Northern blotting is used for the detection of specific RNA fragments. And this technique is called Northern simply because it's similar to the Southern blotting technique. The person who invented this was not named Northern. That'd be kind of a crazy coincidence. Nonetheless, the Northern blot is used to detect gene expression. And so, one way to measure gene expression is through different mRNAs. And so mRNA is a form of RNA. So Northern blotting, think gene expression. Another term you might see is Rt-Pcr, which stands for reverse transcription PCR. So what that does is it takes mRNA and converts it to cDNA, which is complementary DNA, and then that complementary DNA can be sequenced, and again, you can assay gene expression. You can see which genes are being expressed. You can look at differential gene expression, etc. Another term you're going to see is qPCR or quantitative PCR, and this is also called real-time PCR. And okay, and I know what you're thinking right now, and that is, why did some dumbass scientist decide it was a good idea to abbreviate reverse transcriptase PCR, Rt-Pcr, when there is such thing as a real-time PCR? And I fully agree with you. Makes no sense. Stupid. However, just know that Rt-Pcr stands for reverse transcriptase PCR, not real-time PCR. However, they kind of measure the same thing. You're looking at gene expression. They both do that. So if for some reason you couldn't remember that on the Mcat, probably not the biggest deal in the world, but worth mentioning. So how exactly does qPCR help you look at gene expression? Well, qPCR basically allows you to watch a PCR reaction as it goes. So you say you have some mRNA species that you want to look at, quantify it, how much of this gene is there. Well, if you know its sequence and you can design primers to bind to the ends of it, then you can do a PCR reaction. So say you have those primers, binds to your gene, your mRNA, your cDNA, and it will start to amplify that. Well, the qPCR also has some kind of fluorescent dye that it's using, and so usually this is a probe, and so your probe binds somewhere on this DNA sequence, and as your polymerase goes across, it hits that probe, and that probe fluoresces. So over time, you're going to get more and more fluorescence, and you can track that fluorescence. And so that fluorescence then corresponds to some concentration of DNA. And so then you can use standards and you figure out what that concentration or the original concentration of your gene was. Boom. And that's how you measure the amount of gene that you have in a sample, qPCR. But again, the most important thing to know is that for real-time PCR or qPCR and reverse transcription PCR, you are looking at gene expression. And same goes for northern blotting. All right, the last molecular biology technique that applies to genetics that I want to talk about is FISH, or fluorescence in situ hybridization. So the purpose of FISH is basically to see where in a chromosome a certain sequence lies or certain sequences lie. It's a pretty simple concept, really. What you do is you throw in a probe that binds to a particular region of a chromosome, and that probe fluoresces, and then you can literally image it and see where it's fluorescing from. And so it's useful, for example, to help a researcher or clinician identify where a particular gene falls within someone's chromosome. The first step then is to prepare short sequences of single-stranded DNA that match a portion of the gene the researcher is looking for. So this is like a primer, right? Because you have to know the DNA sequence before, and that way you can kind of target. So this is how you target. You know the sequence before, and then you create that single-stranded sequence called a probe. The next thing that the researcher would do then is attach some kind of fluorescent dye to that probe so that it can be seen, and then you mix that probe in with your DNA template or your chromosomes, and then the researcher is actually able to visibly see where on the chromosome that genetic sequence lies. All right, so real quick recap of everything I just talked about. So PCR is used in order to amplify a certain sequence. And, you can either know that sequence and design primers for it, or you can just amplify a bunch of, whatever the hell you want to amplify, the whole genome. And in that way, you don't need specific primers. And then you have these different blotting techniques: Western, which is for proteins; Southern, DNA; and then Northern, RNA. And another thing that Northern is good for is measuring gene expression. And then you have reverse transcription PCR, quantitative PCR, which is the same as real-time PCR. And these two methods are used to measure gene expression. And then lastly, you have FISH, or fluorescence in situ hybridization. And it is used in order to see where in the chromosome specific sequences lie, specific DNA sequences lie, that is. And all right, with that, we are done with the 2-part genetic series. And this has got to be probably the longest topic I'll talk about, but obviously very important, or else I wouldn't have spent so much time and effort in making these 2 podcasts. But trust me, listen to these, know all these different terms, definitions. It'll definitely help you on the MCAT. So hope this helped, and listen to the next podcast. I think I'm going to start getting into some sociology and psychology topics, which is a little bit different. I prefer the science stuff, but that's all right. All right, now for the MCAT Advice of the Day segment. So my advice for you today is pretty simple, and that is spend more time than you did taking practice tests, and also practice materials that you spent doing them. Spend more time analyzing, why you got questions wrong, why you got questions right. What is the MCAT trying to have you think? So spend more time analyzing the work that you do than actually doing the work. I think it's easy to sit down and say, okay, I'm going to do 2 tests a week. I'm going to bust this stuff out. It's going to be great, which that is great. Taking a lot of practice tests is awesome, but make sure you spend a lot of time actually reviewing what you did and making sure you know why you're getting questions wrong. This podcast was written and produced by Murph the Dog. Music and other guest components were also produced by Murph the Dog. Thank you for listening.